Hello, I'm Reagan Bain, and today I'll be presenting my research conducted under Dr. Amanda Williams, focusing on the determination of apoptosis in colorectal epithelial cells upon treatment with HG5. This project is part of a larger project that examines different parts of inflammatory bowel disease. But what exactly is inflammatory bowel disease? Inflammatory bowel disease, or IBD, is a chronic inflammatory disease characterized by the inflammation of tissue of the gastrointestinal tract. According to the CDC, 1.6 million Americans are currently living with IBD. Some of the most common symptoms are abdominal pain, bloating, and diarrhea. There are several different forms of IBD, the most common two being ulcerative colitis and Crohn's colitis. For ulcerative colitis, inflammation is continuous and occurring mostly in the colon and rectum, as you can see here highlighted in the picture on the right of the screen. In Crohn's disease, which is the focus of this project, inflammation occurs in patches along any part of the GI tract. And unlike UC, which occurs in the mucosal layer, Crohn's disease can occur in multiple levels. In Crohn's colitis, inflammation only occurs in the colon. In this project, all cells were treated with human alpha defensin 5. HG5 is an antimicrobial peptide that is secreted by Paneth cells that are normally located in the small intestine. HG5 functions to regulate pro-inflammatory cytokines, and it leads to cell death by binding to and disrupting cellular membranes and secreting IL-8. Previous testing by Dr. Williams revealed an abnormal presence of Paneth cells in the colons of Crohn's colitis patients. This study also found that human alpha defensin 5 was upregulated in the colon of patients with Crohn's colitis, but not in ulcerative colitis patients. This led to an experiment completed in our lab previously that revealed upon treatment with HG5, colorectal epithelial cells experienced cell death. The image on the right of the screen shows the data from this study. As you can see, cell death occurs mostly at the 8-hour and 12-hour time periods. These cells were treated with 0.5, 1, and 2 micrograms of HG5. Yet, the exact mechanism of cell death is unknown. This study I'm presenting looks to explore a possible cell death mechanism. For this study, we hypothesized that a mode of cell death occurring in human colonic epithelial cell line MCM 460 is conical in nature, which will reveal evidence of active caspase 8. In order to conduct this experiment, we used standard culturing techniques with M3 base A media on our NCM 460 cells. Cells were treated with 0.5 and 1 micrograms of HG5. Protein was extracted using the per mammalian cell thermo-kit procedures at an 8 and 12-hour post-treatment. An SDS page and Western blot were conducted using caspase 8 as the primary antibody and the mouse as a secondary antibody. Shown here on the screen is the result of a Western blot. As you can see in this top line is the banding for pro-caspase 8, which is our inactive caspase, and it appears around 55 kilodaltons. The bottom band, which is a little fainter, is a band for our cleaved caspase 8, which is our activated caspase 8, and it appears around 10 kilodaltons. The first 3 samples past the ladder are 8 hours, and the remaining 3 are 12-hour time period samples. This statistical breakdown you see on the screen highlights what we saw from the plots. The image on the left of the screen shows the levels of pro-caspase 8. This is the inactive form. The image on the right shows the levels of cleaved or activated caspase 8. As you can see in both images, the levels of caspase is higher in our 8-hour samples. This just means that overall we have a higher expression level of caspase 8 in our 8-hour samples as compared to our 12. However, when we look at them both together, we really don't see any significant difference between the two. The figure on the screen here shows that the ratio or percent of active caspase in each sample. This data reveals that the amount of activated caspase 8 is higher in the 12-hour samples. No significant difference is seen amongst the 8-hour samples. Another project being conducted in the lab looks at the activation of caspase 1. As you can see from the figure on the right, there is no change in the levels of active caspase as compared to control. And this is true for your 8 hours and your 12-hour samples. As you can see in mine, there is an increase in our 12 hours. So to us, this can suggest that maybe caspase 8 does play a role in a cell death that is occurring in the cells we are seeing. In conclusion, these Western blots revealed a presence of activated caspase 8 in both the 8 and 12-hour samples. We saw an increase in the presence of activated caspase 8 in the 12-hour samples as compared to the 8-hour. However, further experimentation is needed to really confirm what we saw today. For future directions, as I stated, I'd like to complete more studies and do more Western blots to really confirm the results that we've seen. This data shown today was with 2 completed blots. A third blot was conducted, but was unable to be used in this presentation. A third blot could help reveal any significance in the data or any differences in the data. I would also like to rerun the blots using a higher concentration of the primary antibody. These experiments were conducted using a 1 to 500 dilution. I would like to increase that to a 1 to 200 dilution. This could maybe help clarify the banding. Some of the blots were very faint, and others we had multiple bandings that seemed to run right into each other. So using a higher concentration could possibly help bring out the bands more intensely. The data sheet for our primary antibody also stated that the active caspase would appear around 10 kilodaltons, as ours did and was shown in this presentation today. However, images of blots attached to the data sheet showed the cleaved caspase around 40 kilodaltons. So with a higher antibody concentration, we can maybe get a clear definition of banding and possibly be able to clearly see what is activated and what is cleaved and not activated. There are also many different types of cell death. And maybe what we see isn't apoptotic, maybe it's necrotic, but comparing to other studies would give us a clear insight to exactly what is occurring. There are other studies in our lab being conducted with a general caspase inhibitor, and from there, comparing my studies to hers, we could possibly understand a deeper mechanism or insight into the apoptosis. I'd like to thank you all for listening. Thank you to Dr. Amanda Williams and Hannah King for your guidance and help. Thank you to the Lipscomb Biology Department for funding the research and for the opportunity to learn.